hormones asking the ovaries to produce more than one secondary oocyte. So the doctor will just get several, let's say 10, or successfully get 10 secondary oocyte from this woman. Normally it won't, okay, just because of the hormone injection. And then from a man's sperm, just collected, and they would just artificial to perform in the laboratory. They try to merge, do the fertilization with the egg and the sperm. Okay, so it has been invented many years ago, several decades ago, and then they gained Nobel Prize in 2010. So later on, people even use another method, which is also belong to IVF. That is intracytoplasmic sperm injection. The doctor can, or the scientist can just inject the sperm directly into the egg cell. Okay. Okay, so far any question? So pregnancy, after the egg has been fertilized, of course, the woman got pregnant. So in day 0, we say that the day of the fertilization is day 0. On day 0, after day 0, it will start to have it become a zygote. The fertilized egg becomes a zygote, and then this zygote will start to do mitosis, continuously mitosis until within a week. So you can imagine within a week, this cells group we call blastocyst. Within a week, this blastocyst will move from the uterus, from the oviduct to the uterus, right? And then they finally can attach onto the uterus wall. So this is the reason why, if you still remember just now when we're talking about the menstrual cycle, the progesterone and the estrogen are kept high. They're produced high and then reach the maximum within a week, right? That is because just preparing this week to allowing the blastocyst to arrive on the endometrium of the uterus. Okay, so for days 7 to 10, implantation happens in the uterine wall. So the babies will just sit there. So just a reminder here again, on the 6th day after the fertilization, the trophoblast cells will secrete some enzyme, allowing the blastocyst to sit onto the uterine wall. So during the implantation, if you still remember, at this moment, the blastocyst itself releases a hormone called human chorionic gonadotropin, or in short term, HCG. This hormone, just like LH. If it is just like LH, they can just mimic the LH function. So to keep the corpus luteum continuously to be alive, we don't want the corpus luteum to degenerate because they are keeping on releasing estrogen and progesterone. Okay, and this keeps the whole thing, the endometrium, thick and increase the vascularization to house the blastocyst. That means not only the tissues being thicker, also more blood vessels can be seen. Okay, and then this HCG hormone is interesting. You can see in the first 3 months of the pregnancy, the HCG hormones will be produced at maximum. What does it mean? This is one, okay, internally, it is asking the corpus luteum to be alive there. Okay, secondly, it is letting the pregnant woman to know that she got a baby. How to test it? So you already know that the rapid test, right? The rapid test now to test COVID-19. Some people would just say that, oh, they're just like a pregnancy test. Yeah, because they are using the same mechanism. So that's why they look like very similar. Okay, so what they do is to test the HCG in, this is the spelling mistake, HCG in the urine of the pregnancy woman. Okay, usually it can be tested, it can be detected even shorter period of time, maybe 7 days, as short as 7 days. But usually they wait for 1 month, okay, or half month, and then to test it. So urine or serum containing the hormone, if the level is high enough, then it can be very accurate. Okay, so that is, so everybody know what does it mean by positive now. So I asked you a question. I finally think about one question related to your professional. Do you know when will the embryo develop their eyes? The embryo develop the eyes. In which week? starting from which week? Can you guess? Do you know that? The embryo developing the eyes. Okay, I answer myself. It is starting from week 3. Week 3. So you can see that the babies develop their eyes quite early and start from week 3 and then develop until week 10. Right, around this period of time, the early period of the development of the embryo. Okay, this is the only related thing. So then the embryo in week 5, you can see many things like the umbilical stalk has already been developed. And then you can also find the placenta is well developed already. The yolk sac, the chorionic villi of the placenta, everything has been well developed, and we can already see the babies clearly. Okay, the embryo clearly. And until the week 10, you can already see the whole baby shape already. And then the umbilical cord can be seen. Placenta providing the nutrients. Okay, amnion and the amniotic cavity. And inside, you can find the amniotic fluid. Okay, and the chorion. So then the mothers, they need to check whether the babies have any abnormalities. Not only using the ultrasound, we can test the baby shape, okay, appearance, or development of the limbs, of the head. We can use ultrasound. But how about the genetic problem? So before 2011, there is a technique called amniocentesis. Amniocentesis is the test that we use. You can see from the picture here. We use the ultrasound to find the baby, where the baby locate, and then we use a syringe, very thick needles, to take some samples from the amniotic fluid. Okay, from the amniotic fluid here. So this is quite invasive but accurate. So they just take some fluid, because inside the fluid, what we can find is the cells sloughed off from the fetus, sloughed off from the placenta, and also the amniotic sac. Okay, all these cells, they having the same genetic composition. So we just take the cell debris there, and then we can get the genetic information, and we will know whether the babies, the chromosomes got any problem. So especially when we are going to test the chromosome 21, that to prevent babies will be developed into Down syndrome. So this is an old method, we can say that. So before 2011, and this invasive method will performed around at about 16th week of the pregnancy. Why we say that this is invasive? Because the risk can be around 1% to hurt baby. Okay, even though the number seems to be small, but no mother would like to have any risk on it. So we have a Hong Kong scientist. I hope you know him. So he is Professor Dennis Low. Professor Dennis Low, he discovered, before 2011, he already discovered that accidentally. He found that we do not need to do such kind of invasive test to test the baby genetic problem, because he discovered that if we extract the blood from the pregnancy woman, we can get the cells from the babies as well. So that is really interesting, because in the past, the placenta, how they work is they're preventing the blood from baby to mom and from mom to babies, right? But then they still find that there are some cells from the baby appearing in the mother's blood. Should be some DNA, more precise. Okay, some DNA from the baby appearing in the mother's blood. So they just get a DNA there, and then go to check and find out, wow, amazing. This type of very easy method, no invasive at all, no harm to the baby at all, and they can get the data. And this data nowadays have been applied in over 90 countries, using this method for the baby testing. Not baby testing, not good word, for the analysis of the pregnancy, of the fetus. Okay, so this method has been used in Hong Kong for some years already, more than a decade, and then yeah, so This method is very useful. So exchange molecules across the placenta. What we just want to prevent the materials from the babies getting to the mom, the blood from baby to the mom, and vice versa. So umbilical arteries deliver the fetal blood to placenta vessels, and they can exchange the materials or molecules inside. So I don't want to repeat this slide. This is just a reminder or revision. So the main thing is what you need to know is